Namaste students, Kushro Vidya. I, JV and Dr. Chandrakala, Assistant Professor at Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the control of the cell cycle. The length of the cell cycle is variable even within the cell of a single organism. In human, the frequency of the cell turnover ranges from few hours in every embryonic development to an average of 2 to 5 days for epithelial cells, and an entire human life time spent in the G0 by specialized cells such as a cortical neuron or a cardiac muscle cells. There is also a variation in the time that a cell spends in each phase of the cell cycle. When fast dividing mammalian cells are grown in the culture, growing the length of the cycle is about 24 hours. In rapidly dividing human cells with 24-hour cell cycle, the G1 phase lasts approximately 9 hours and the S phase lasts for the 10 hours. The G2 phase lasts for about 1.5 hour. The M phase lasts approximately 1 hour. So in early embryos of the fruit fly, the cell cycle is complete in about 8 minutes. The timing of the event in the cell cycle is controlled by the mechanism of both external and the internal cells. So both the initiation and the inhibition of the cell division are triggered by the event of external and the cell when above begins the replication process. It is essential that the daughter cells produced by the exact duplication of the parent cell and the mistakes in the duplication or the distribution of the chromosome lead to the mutation of that may be passed from forward to every new cells produced from the abnormal cells. A checkpoint is one of the several points of the eukaryotic cell cycle at which the progression of a cell to the next state in the cycle can be halted until the conditions are favorable. So the G1 checkpoint determines whether all the conditions are favorable for the cell division to proceed. The G1 checkpoint are also called the restriction points and is a point at which the cell irreversibly commit to the cell division process. The external influence such as the growth factors plays a large role in carrying the cell past in the G1 checkpoint. In addition to the educate, reverse, and the cell side, there is a check for the genomic DNA damage at the G1 checkpoint. So the G2 checkpoint is a bar entry of the mitotic phase if the certain conditions are not met. As the G1 checkpoint, the cell size and the protein reverse are accessed, or the G2 checkpoint is to ensure that all the chromosomes have been replicated. So the M checkpoint occur near the end of the metaphase and the stage of the karyokinesis. So the M checkpoint, also known as the spindle checkpoints, or because it determines whether all the sister chromatids are correctly attached to the spindle microtubules. So in addition to the internal control checkpoint, there are two group of intercellular molecules that regulate the cell cycle, and these regulatory molecules, or can say that, and either promotes or progress the cell to the next phase and halt the cycle, or a negative regulation. A positive regulation of the cell cycle is the two group of the protein called the cyclin and the cyclin dependent kinase are responsible for the progress of the cell through various checkpoints. So the cyclin regulates cell cycle only when there is a tightly bound uh, cyclin complex formation or the cyclin bind to a specific point of the cell cycles or a cyclin complex proceed through the checkpoints, and here many of the inhibitory molecules directly or indirectly monitor the particular cell cycle events. The negative regulatory cell cycle, where the second group of a cell cycle or a molecule negatively regulate and all the cell cycle. The member of that a positive regulation of the active molecules can cause the cell to progress the bind. The best understood negative regulatory molecules are retinoblastoma and the protein RB of B53 of B21. Retinoblastoma proteins are a group of tumor suppressor proteins common in many cells. So the 53 and 21 designates refer to the functional molecular mass of the protein P in kilodalton. The cycle comes from the research conducted which have a lot of regulatory controls, and all the three of the regulatory proteins are discovered to be damaged or non-functional in cell that begins to replicate uncontrollable. In case many cause unchecked progressions in the cell cycle or a faulty copy of the regulatory proteins. The RB or a P53 or P21 act primarily as G1 checkpoint, and the P53 is a multifunctional protein that has a major impact on the commitment of a cell division because of it act as to damage the DNA in cells that undergo a preparatory process during the G1. If damaged, the DNA is detected. P53 halts the cell cycle and recruits the enzyme to repair the DNA. If the DNA cannot repair, the P53 can trigger apoptosis and a cell suicide to prevent a duplication of the damage of a chromosome. So here the P53 level rises and the production of the P21 is triggered, and P21 enforces the halt in the cycle dictated by the P53 by binding to the inhibiting of the activity of the gliadin and exposed to the more stress. So here the higher level of the P53 and P24, 21 accumulate and make it less likely, and the cell will move to the S phase. The RB exists the influence of the positive regulatory protein. Okay. So the growth of the cell triggered by the phosphorylation of RB, where the phosphorylated RB release the E2F, which binds to the DNA and turns the gene expression and thus advancing the cell cycle. So the each steps in of the cell cycle is monitored by an internal control checkpoint, where there are three checkpoints in a cell cycle. That is one near the end of the G1, and the second in the G2 by M transition, and the third is in the during the metaphase. So the positive regulatory molecules allow the cell cycle to advance in the next stage, whereas the negative regulatory molecules monitor the cell's conditions and can halt the cell until a specific requirement is met. Thank you. This session is powered by 2.0 Vidyapeeth Women's University, Jaipur. If you have any queries, we'll discuss in the problem solving session. Thank you.